Hi everyone! Today we are looking at sex linkage, and more specifically, we are looking at X-linked genes. Here we see that hemophilia is an X-linked recessive disorder in humans, and we are asked to determine the phenotypic and genotypic proportions of the resulting progeny for the following crosses. So, let's start with the cross in part A and designate some representative alleles for the individuals in this cross. Since we know that this is an X-linked disorder, we know we are looking at the sex chromosomes, where females generally have two X chromosomes and males generally have one X and one Y chromosome. If we designate big H as our dominant allele and little h as our recessive allele, we can then add these alleles as a superscript on these sex chromosomes. So, the woman in this cross has hemophilia, and since this disorder is recessive, it means that she has two recessive alleles for the hemophilia gene. The man in this cross, on the other hand, does not have hemophilia. As a result, he has the dominant allele for this gene. Notice that I put the allele only on the X chromosome, as this is an X-linked disorder, and therefore there is no allele on the Y chromosome. This means that for X-linked genes, males will only have one copy of the gene and thus will only have one allele. Now that we know the genotypes for the parents of this cross, we can then determine the genotypes for the progeny using the Punnett square seen on the left. We can place the parental alleles on the top and sides of the square, and then determine the genotype for the progeny using the remaining 4 squares. Alright, so based on the Punnett square, we can see that half of the progeny will have this genotype, 100% of which will be female, and the other half of the progeny will have this genotype, 100% of which will be male. As for the phenotypes, again, we know that this is a recessive disorder, and so for a female to be affected by the disorder, she will need to have two recessive alleles, one on each of her X chromosomes, while a male who only has one X chromosome will only need one recessive allele to be affected. As a result, we can see that half of the progeny will be unaffected by the disorder, 100% of which will be female, and the other half of the progeny will be affected by hemophilia, 100% of which will be male. Alright, so in part B, we would apply a similar approach. So again, designating representative alleles for the parental generation. So a man with hemophilia will have an X and Y chromosome, with the X chromosome possessing a recessive allele for the hemophilia gene, and a woman who is a carrier for the hemophilia gene will have two X chromosomes, one of which will contain the dominant allele for hemophilia, and the other will have the recessive allele. Now that we've determined the parental genotypes, we can put them in a Punnett square and determine the genotypes for the resulting progeny. So, we can now see that there are 4 different genotypes that appear as a result of this cross. 1 out of the 4 resulting offspring will have this genotype. 1 out of the 4 will have this genotype, and a quarter will have this genotype, and the last quarter will have this genotype. And so, based on the Punnett square, we can determine that there is a 1 to 1 to 1 to 1 genotypic ratio for these genotypes, compared to part A, where there was a 1 to 1 genotypic ratio. As for the phenotypes, similar to part A, we can determine that half the progeny will be unaffected and half of the progeny will be affected by hemophilia. So, Based on this, we can see that the phenotypic proportions are the same, but the genotypic ratios differ depending on which parent has hemophilia and if the other parent is a carrier for that gene or not. Alright, that is it for this video. Thank you for listening, and we'll see you in the next one.